Friends, topic of today is an approach to the diagnosis of lymphoma. The topic is taken up today because in case of lymphoid tissue, it is difficult to differentiate benign from malignant condition due to heterogeneous appearance. Proper steps should be followed for diagnosis. A multidisciplinary approach is required. Regarding morphology, there are three parts of lymph node: cortex, paracortex, and medulla. Cortex has follicles, primary and secondary. Primary without germinal centers, secondary follicles with germinal center. Germinal center has dark zone formed by centroblast and light zone formed by centrocytes. This type of polarization indicates normal or reactive process. So germinal center has centroblast and centrocytes, B lymphocyte in origin, scattered follicular T helper and regulatory cells, tingible body macrophages, and follicular dendritic cells. Mantle zone is tightly packed small B lymphocyte around germinal centers. Marginal zone is lighter zone on outer rim of mantle zone formed by less packed small B lymphocytes with more cytoplasm. They are mix of post-follicular memory B cells and naive B cells. Naive B cells are B cells without antigenic exposure. Paracortex is the tissue between the cortex and medulla. Contains predominantly dark staining mature T cells, B immunoblast, interdigitating dendritic cells, plasmacytoid dendritic cells, histiocytes, and high endothelial venule. Medulla is near hilum, has medullary cord, sinuses, and vessels. Medullary cord lie between the sinuses, has mostly small B and T lymphocytes, plasmacytoid lymphocytes, plasmablast, and plasma cells. Sinuses are lined by macrophages, mast cells, and plasma cells. About 70 entities are there in WHO classification of lymphoid neoplasm. Broadly, we can divide lymphoid neoplasm into lymphoma, which may be non-Hodgkin's lymphoma or Hodgkin's lymphoma. Non-Hodgkin's lymphoma may be of T or NK cell or B cell in origin. Tumor may arise from precursor or mature B or T cell. Other lymphoid neoplasms are immunodeficiency associated lymphoproliferative disorders and histiocytic and dendritic cell tumors. All the steps should be meticulously taken care of for good ultimate result. Clinical information, excision, transport, fixation, processing, sectioning, and staining. Transport, fixation, processing, sectioning, and staining will result in good hematoxylin and eosin slide depicting fine cellular details. This will help in correct provisional diagnosis with differentials. Regarding clinical information, age, sex, site of lesion, previous illness, medication like steroids or other immunosuppressive drugs, immune status of the patient like HIV status, extent of involvement, CBC bone marrow findings, and history of autoimmune disease are important. Regarding sampling, intact lymph node should be sent to the lab without crushing. It should be sent in wide mouth bottle. 10% neutral buffer formalin is used as a fixative. The tissue should be sliced immediately for proper fixation. Fixation time is 12 to 36 hours depending upon the size of the tissue. One should avoid both under fixation and over fixation. RPMI medium for flow cytometry, stress tissues for cytogenetics and flow cytometry. Methodical microscopic examination of lymph nodes should be done concentrating on each areas like capsule, subcapsular sinus, cortex, paracortex, sinuses, medulla, and vasculature. Scanner view to see the architecture. for effacement, partial or diffuse, to detect pattern like diffuse, nodular, follicular, mantle zone, marginal zone, or sinusoidal. Starry sky pattern is there or not. Difference between nodular and follicular pattern. In nodular, nodules are big, outline blurred, not well defined. In follicular pattern, small distinct nodule. In high power, one should start identifying cells to differentiate neoplastic from reactive process, inflammatory, necrotic, fibrotic, and granulomas. If neoplastic process is there, so non-Hodgkin's lymphoma or Hodgkin's lymphoma. If non-Hodgkin's lymphoma, mature or immature cells are present or not. Large cell, intermediate cell, small cell, cleaved or non-cleaved, mixed cell, blastic cells, Reed-Sternberg cells. One should see for non-neoplastic cells in case of neoplasm. These are the cytological features of different types of cells. Immunohistochemistry is used for confirmation of diagnosis, for identifying cell lineage and phase of maturation, detecting specific genetic alteration and degree of cell proliferation. Also for identifying therapeutic targets like CD19 and CD30. It is also helpful in prognosticating the case. B cell development stages has molecular and immunophenotypic biomarkers which are used in the identification of tumors. Similarly, in case of T and NK cell developmental stages, one should know, as different stages have molecular and immunophenotypic biomarkers used in the identification of tumors. Regarding planning immunohistochemistry, one should interpret hematoxylin and eosin slides and make a differential diagnosis. Then one should proceed for immunohistochemistry. These are the different immunohistochemistry panel for different cell types. These are the normal immunophenotype for different cells. These are the essential immunohistochemistry markers for different types of cells. These are recommendations for T or NK cell lymphomas, recommendation for diffuse large B cell lymphoma and its variants, recommendation for small or low grade B cell lymphomas, recommendation for precursor lymphomas, recommendation for Hodgkin's lymphomas. Immunophenotypic algorithm, the HANS or TALLY, are used to predict the cell of origin in cases of diffuse large B cell lymphoma, like the cell of origin is germinal center B cell or activated B cell. Each is approximately 80% concordant with gene expression profiling results. If in doubt, one should go for molecular studies like fluorescent in situ hybridization on paraffin blocks for translocation and polymerase chain reaction on paraffin blocks to prove monoclonality, like T cell receptor gene rearrangements. Regarding T cell gene clonal rearrangement, T cell receptor either alpha, beta, gamma, delta are produced following somatic rearrangement of T cell receptors genes like alpha, beta, delta and gamma. This process is vital to proliferation of T cells in normal immune function, but can be exploited to aid in the distinction between reactive and neoplastic processes. A reactive benign T cell proliferation is characterized by polyclonal expansion of T cell, whereas a malignant process is characterized by clonal expansion, that is identical gene expression profile of one T cell population detected by polymerase chain reaction. This can be used to aid in the diagnosis of malignant T cell lymphoma or to measure minimal residual disease. Regarding methodology, by extracting genomic DNA from blood, lymph node, bone marrow, or other tissue types, T cell receptor gamma and T cell receptor beta are amplified by polymerase chain reaction. Precise fragment sizing of the amplicons is accomplished using capillary gel electrophoresis. The presence or absence of a monoclonal population is determined based on the overall assessment of the gel electrophoretic pattern. The specimen requirement for this process is either blood 5 to 10 milliliter or bone marrow aspirate in case in citrate or irritate tube, formalin fixed paraffin embedded tissue blocks, fresh tissue suspended in sterile culture media. Molecular studies processes are flow cytometry or gene expression profiling. Flow cytometry detects clonality, abnormal light chain restriction. In case of B cell non-Hodgkin's lymphoma, chronic lymphocytic leukemia and hairy cell leukemias, this is used for relative quantification, abnormal phenotype, definitive diagnosis, and in case of follow-up and to detect minimal residual disease. Regarding gene expression profiling, a laboratory method that identifies all of the genes in a cell or tissue that are making messenger RNA. Messenger RNA molecules carry the genetic information that is needed to make proteins from the DNA in the nucleus of the cell to the cytoplasm where the proteins are made. A gene expression profile may be used to find and diagnose a disease or condition or to see how well the body responds to treatment. Analog of gene expression profiling is glossary barcode that differentiate different types of glossaries. One can know about therapeutic response. Methods of gene expression profiling are either microarray, that is DNA chip, or next generation sequencing. Use of molecular studies is in definitive diagnosis, definitive subtyping, in prognostication, and in follow-up of cases. WHO lymphoma diagnosis requires the combination of various techniques like morphology, immunohistochemistry, T cell receptor gene rearrangement, fluorescent in situ hybridization, and rarely electron microscopy. A few words about double hit and double spatial lymphomas. Double hit lymphomas, two recurrent chromosomal breakpoint aberrations used to be found and indicates bad prognosis. In case of double expression lymphomas, co-expression of BCL2 and c-MYC on immunohistochemistry, and this also has poor prognosis. Thank you.